Hello friends, welcome to part 2 of lecture 2 of week 11. So in the first part we discussed the programs and tools that we can use to predict genes in prokaryotic genomes. In this part we will look at the prediction of other kinds of ribosomal RNA genes, RNA genes basically including ribosomal RNA, transfer RNA, etc., right, and operons also, and also how to annotate the genes that we have predicted, the protein coding genes in the first part. So first we will start with the annotation of the genes. So basically we want to annotate the gene because genes are performing functions which are essential for the genome, and knowing that function will help to understand the genome better. It could be a part of the pathway, could be a drug target in case of pathogenic bacteria, or may also be a commercially useful, or that can be used in molecular biology or other applications. So then when you do the annotation, you may find that some gene function you will know by BLAST or AGNOG or other methods. So you can say that okay, this is the function of this gene and this is a known function gene. There could be some genes for which you will have a putative function, that means predicted function, that okay, this may do this kind of a job, that you will find by other programs like using motifs or fingerprints or interproscan, those kind of programs. It could be a hypothetical gene, that it is a proposed gene and basically it is having the features of the gene but its function is not known, right? Okay, and it may be considered later for further experimental or computational analysis. Considered hypothetical are those which are genes which are identically present in multiple genomes, but still their function is still unknown. Okay, so they can be single genes with unknown function, or there could be genes which are present across genomes, and that means they definitely are doing some function, but they are unknown, right? And the tools that you may like to use are BLAST, that we also did for eukaryotic but also good for prokaryotic, operon-mapper, interproscan, ghost koala. Okay, so this we will discuss today. So BLAST is one application that can be used to predict the function of the genes. You can upload the file in the FASTA format file in terms of nucleotides or proteins, and specify the database NR, or basically a particular genome, or those kind of databases. Okay, specify that what kind of you want to do, fast search or slow search and like that. Okay, and give the other parameters like generally default is always good. Okay, and then when you say submit, it will give you a list of annotations for a given set of queries, right, or a single query, like your protein or genome sequence matched to this RNA. So it was a nucleotide sequence. It has a very good bit score and maximal score, and basically the bit score was high, total score also high, query coverage 100%, identity 100%. That means you have found a kind of known gene for this. So basically this way BLAST can be helpful in finding genes, function of genes for which something like that already exists in the databases. Another approach is COGS, or cluster of orthologous groups, so in which an effort was made to classify the proteins into orthologous genes, set of orthologous genes, and each group or cluster of those genes can be assigned with one kind of function, and broadly those functions can be regrouped into a broad function like information or metabolism or translation, okay, or signaling. Okay, so basically this gives an overview of what kind of functional classes are more abundant in a genome, and also like what is the exact annotation for that gene also. So COG classifier is one program that was developed by NC and it is still there, right? And the COG, these are the different COG classes. So you can see that RNA processing and metabolism, labeled as A class, is basically a broad class and does not indicate a single gene but a group of genes that are doing this function, this kind of function in the cell, right? So similarly for E. coli, if we run this, so you will find that G appear to be the most abundant class, G, E and C, which are carbohydrate transport and metabolism. amino acid transport and metabolism, energy production. So basically these functions are more abundant in E. coli as compared to others. So this gives you some kind of idea of what that genome is and what kind of functional classes are abundant in that genome. Okay. Now based on COG, AGNOG developed used those COG classes and developed a program called operon-mapper, okay, which also does the orthology based assignments, and also in addition to that it also gives you the KEGG annotation, geo annotation, and pathway information. So basically it's very comprehensive in like annotating the function, and that is why it is a method of choice. It may do lesser prediction as compared to BLAST, but it will give you a lot more information compared to BLAST, and basically combination of the two can also be used, and also it is good for the metagenomic proteins. Okay, so this is the operon-mapper, basically for which can take your set of sequences or genome or proteins. It will run prodigal to find the proteins, and then it will use the its own database of AGNOG, okay, to do a quick search by using HMM diamond or MMSeq2, okay, and then it will try to arrange that into the orthologs and then classify into various functional categories and provide the annotation. Okay, so this is the functioning of operon-mapper which we just discussed. So this is the web interface for operon-mapper. It is good to run online also because it can take unlimited input, and a lot of proteins, which is always more than what you can provide generally, right? So you can give protein sequencing, CDS, genome, metagenome, or seed annotations from mgrast and upload the file, okay, and submit it, and it will give you a comprehensive details of the genes that you had, e-value and score for that, the annotation or AGNOG ID, the exact functional annotation, right, gene symbol, GeoIDs, EC numbers, KEGG IDs, okay, KEGG pathway. So all this information you can get in a single go by using the operon-mapper. Okay, then geo number that we saw like in the previous slide. So gene ontology is the process of classifying a gene into 3 different groups based on its function in the cell. So it could be a molecular function, right, that tells that what exactly that protein is doing, like catalysis or transport or signaling, okay. It could be a cellular component, that in which part of the cell that gene is doing that function, right, and also the biological process or the larger process in which, like metabolism, in which that gene might be doing the function, okay. So these 3 informations are very helpful in further annotating the gene, and this you can get by operon-mapper. Similarly, the operon-mapper also give you the KOID or the KEGG information. So KEGG's database is a reference database for pathways, genes, reactions, ligands, substrates, okay. So it's a very comprehensive updated database where you can find information about all the pathways that may exist, metabolic pathways, their implications in diseases, the gene candidates, okay, the exact reaction, the exact metabolic reaction, the substrate, its products, okay, the pathways in which those reactions can map to, the genomes in which it may appear, right. So this is very important for a new genome because it has already the reference pathway, and you can map your genes against those pathways, and this information can be generated by operon-mapper and also by the KEGG tools, right. So it's a very comprehensive database and highly useful database. So it has so many information like pathway, modules, orthology, genes, genomes, classes, okay, diseases. So it's a very comprehensive database that is used for the metabolic pathway annotations, right. And KEGG has several parts, of which like KO or KEGG orthology may be directly helpful in terms of annotation, because it is like a GeoID or an AGNOG ID which tells you about the function of that gene, right. And each KO ID has a corresponding AGNOG ID and also a like corresponding GeoID, so that helps you to quickly map it across the various annotation platforms. So for a detailed KO ID you can find for the information such as for this, okay, what is the name of that particular KO gene or KOID or annotation, the pathways to which that will belong to, okay, and then the reactions it is performing, and basically in what disease that KOID is implicated, right. So these are very helpful and information which is pre-compiled, and you can use that to do the functional interpretation of your gene, okay. Now this is a reference pathway of KEGG, and it has, you can see several genes which are represented like as EC numbers or metabolic enzymes, okay, and this is the reaction it is doing, like A to B, B to C, like, okay, from here to here, okay, reaction. So now this is a complete pathway, say steroid biosynthesis pathway. Now your genome may contain a few genes which are, for example, this one, okay, a single gene, or could be multiple genes. So those can be quickly mapped to those pathways, and those numbers or EC numbers will become highlighted, and that can give you a quick overview of any mutation you want to show, or total number of genes you want to plot on this pathway which are present or absent in a genome, and like that, okay. So this is a very, very good way of showing the metabolic or functional profile of genes in genomes. Now also to do this, all this together, there is a pipeline called dFAST, okay, which is published in this paper, and basically it is very fast. In within 10 minutes it can find, annotate genes, also find pseudo genes, okay, also can annotate those genes. So this is a very good pipeline which can take your genomic file, okay, find your assembly gaps, coding genes, RNA genes, tRNA genes, okay, then do the function annotation by using various different approaches, including KEGG, COG, AgNOGs, right, HMM, PFM, T. So basically that is a very comprehensive annotation pipeline that it has, and gives you the output as the in GenBank format, GFF format, and annotated proteins, right. For tRNA, it is using tRNA scan SC, which is a standard program. For RNA, it is using BARNAP or RNA, which is again a standard programs for finding tRNA, RNA, okay. CRISPRs also it can find. So this is one of the pipeline that can be used for quick annotation of a genome, okay. So the example output from this software like is shown here, like this is the length of your genome, okay, total pieces, because you draft genome 26, GC content, okay, N50 of each piece. RNA was 3 genes were, total number of tRNAs were 72, right. So it is a very total number of protein coding genes were 1862. So in just one go you can annotate many things or identify many things in your genome using dFAST. Then if you want to go for individual prediction of tRNA, which are basically very, very important for carrying the amino acid to the protein synthesis site and the ribosome. So different genomes have different numbers of tRNA, and mitochondria also have tRNA, right. A total of 62 should present, like 61 type of genes, total 64 codons are there, so 61 tRNA you expect, but generally it is much lesser than 61 because of the codon degeneracy, okay. So basically tRNA scan software is a standard software, now it is tRNA scan SE, okay, and earlier it was using a COVE algorithm, now it is using Infernal, which does two jobs, which is comparing the tRNA that has found by comparing with the primary sequencing of the tRNAs in the database, because tRNA's form is secondary structure. So it also confirms by predicting that secondary structure and give it a score, okay. So this way it confirms both the primary sequence and the secondary structure, and that it is how like the prediction it makes are highly reliable, right. And basically the new version can also do the mitochondrial tRNA prediction by using the option -M, okay. So you can use it online for smaller genomes, bacterial genomes which are 5 MB or lesser can be easily used, and it can predict the tRNA in those genomes. Or for larger genomes more than 5 MB, you have to download it, right, and by wget this site, unzip it and untar it, go to the folder, configure it for your system, right? Then do make and make install. Make install may require sudo because that is you are doing changes that require root permission. Okay, and the program infernal that it uses requires a separate installation using the same process as tRNA scan. So tRNA scan can be run by using this command, which is tRNA scan -se. -b is like bacterial, tRNA is want to found or eukaryotic or archaeal. So -a, b or k can be used here. -f, that means it is you are also doing the structure along with the primary sequence search. -m for like reporting the statistics of the tRNA finding on the screen, also giving the input file and specifying the output file, right? And also other parameters are also there, but these are the like minimal that you should give. Then it gives a list of tRNA in for your given genome, like the tRNA number and start end, what codon it is meant for, and the score, infernal score, right? And also more information about the structure of the tRNA, and also like how many it found, and what are the pseudogenes of tRNA it found in the genome, and in detail like for what particular codons or anticodons basically it found those tRNA genes. So that is a very comprehensive and commonly used program for finding tRNA scan -se. Now ribosomal RNA, which constitute the major component of the total transcriptome, right? Almost 95 or 90% is only ribosomal RNA. Okay, so because that is why you have to go for ribodepletion if you want to go for the mRNA prediction. So these ones are important to predict, and they are generally in the form of operon that contains the different subunits like 16S ribosomal RNA or 23S or 5.8S, right, in the form of a operon. Okay, so and a bacterial genome may contain more than one operon of these ribosomal RNA genes. So barnap is one of the commonly used program to find ribosomal RNAs in the prokaryotic genomes and also in the eukaryotic genomes, mitochondria also, archaea also. So this is a very comprehensive program. You can download it from GitHub and has a similar way of installation. Okay, so get it from GitHub. Okay, go to the directory, and when you say barnap.help -help, it will display all the possible options that you can use, and like typical command line could be like on the command prompt you will say input file name, okay, that is your genome, then what kingdom it belongs to, like bacteria, archaea or eukaryota. So okay, so like here mitochondria, archaea, bacteria, eukaryota, right? And then the output folder. So basically it needs only information about what kind of ribosomal RNA you want to find, okay, based on the kingdom, right? Then this is the predicted ribosomal RNA sequences in the FASTA format. Okay, so this is one very useful program. Another one is rRNA-mer. So that is also used, but these days people are using either the dFAST or the barnap for predicting the ribosomal RNA. Now operons are one of the important constituents of bacterial genomes, because those that contains genes which are under the common regulation and under a common promoter, and can be expressed transcribed as a like polysystron operon containing multiple transcripts which can be converted into proteins, right? So it is not that all the genes in bacteria are operons. Generally it is metabolic or genes which are doing function in a common pathway or in a common biochemical process, okay, or oral process. So those are likely to be in the operon. So don't expect that the entire bacterial set of genes will be in operon. Okay, so operon-mapper is a program to find the operon in the bacteria and archaea, and it is a open source web server which can be used at this site. Okay, so this is the program where you can give your sequence or upload your sequence, a bacterial genome, and it can, and some annotation for that genome or region in which you found to find the operons, or go for the entire one. Okay, and it will give you the predicted operons. Okay, so basically this lecture was very important to see the how you can annotate the functions to the protein or nucleotide genes that you found in the gene prediction steps in the previous part of this lecture, and how to identify the ribosomal RNA and transfer RNA genes in a bacterial genome, and how to find the operons also in those genomes. So with this we have covered we have completed this lecture, and we have covered the gene prediction, both protein and coding and non-coding genes, right, and also their annotation. So thank you for today.